So here we go again. We are still discussing what is truth. Again, we've gone through the first 5 points already, and we started and are still on number 6, that truth doesn't equal science and science doesn't equal truth. So last week we started discussing some scientific data that focused on DNA. D, you need to pay attention to this one because I know you don't have any background in this. We're going to do more of that today because there's more of this that I want to share with you. Last week we started looking at the structure of DNA, and then I introduced some evidence that we find from that structure. Well, today you're going to get some more evidence from that structure. And I want to begin by just reminding you of a few things that we talked about last week. There we go. It's working. All right. DNA is two covalently bonded molecules running in opposite direction of one another, held together by hydrogen bonds. And this is important because the nucleotides, which were the building blocks of DNA, can only be added to a DNA strand in one direction. They have a front and a back, so to speak, and we did this with our Lego demonstration. Just like Lego building blocks, they won't go together front to front, they won't go together back to back. They have a front and a back, they only go together one way, right? The nucleotides for DNA are the same way. And here's what, hold on, there we go. Here was an image of what all this looks like in 2D. You have, in this image, we have two different strands running in opposite direction of one another, held together in the middle by the hydrogen bonds between the bases. And if you remember, I said there can be millions to billions of these nucleotides in an organism's DNA, depending on their size of their genome and their chromosomes. So this structure right here is what life depends on. Life depends on the ability of DNA to be opened up, have its information accessed, and then closed back up again without being destroyed or altered. And as we said, that happens just like a zipper. The analogy of a zipper is really good here. The DNA always must remain intact and unchanged. If not, the reproducibility found within its structure is lost. Therefore, all its functions are lost. Its information that's mandatory for life is also lost. And the fallout from all that is then life itself is lost. And then we looked at the pattern of bond usage found within DNA, where you have hydrogen bonds in the middle between the bases and covalent bonds everywhere else. And let me go back to this. This in the middle between the bases is what that looks like. Our hydrogen bonds here. All the other bonds holding everything else together, covalent bonds. All right, let's get back to where we were here. Okay. And from that pattern we learn it's not possible for DNA to have come about as a result of randomness or chance. It's not possible because randomness simply won't create the life essential pattern that we see in the bond usage in DNA. Randomness doesn't create patterns. It would create bond usage that's random. And random bond usage would not allow DNA to exist, which will not allow life to exist. And please understand, we're saying all this because randomness is the hallmark of evolution. So that's basically what we went through the other day. So today I want to move on from that. And I'm going to begin by pointing out life depends on an organized, intelligent, logical, choreographed, ordered, whatever you want to call it, pattern of bond usage. The correct placement, the pattern of each hydrogen bond and each covalent bond in DNA. And that's exactly what you find in the structure of DNA. And such an organized, intelligent, logical, ordered, choreographed pattern of bond usage absolutely requires both intelligence and prescience during the creation of the structure. And that takes us to what is called the design inference. And the design inference comes from Dr. William A. Dembski, his book called The Design Inference. The presence of an improbable object or occurrence and the presence of patterns are the two things that indicate both prescience and intelligence were used in something's creation. These two things infer design and the intelligence involved in the creation of that design. And that only comes with a design. And when you have a design, guess what that means? That means there's also a designer. It doesn't come from randomness. And since we have both patterns and an improbable object when we look at DNA, we know that intelligence and prescience were required to create it, because that both, it meets the criteria, both the criteria for being designed. So I want to give you some more examples of the design inference. You have to know and understand all the properties of both a hydrogen and a covalent bond. You have to know their differences. You have to know where each can be used in the structure of DNA in order for it to function as necessary. You have to know all of this ahead of time in order to plan to strategically, intentionally, and intelligently use them in the design and creation of DNA. Randomness, again, the basis of evolution, does not create organization, intelligence, logic, choreography, patterns. Doesn't create any of that like we find with the bond usage. Randomness, by definition, is the opposite of organization, intelligence, logic, choreography, and patterns. It doesn't result from prescience or intelligence because it's random. I think maybe I skipped one here. There we go. There's the one we're on now. The opposite of. That's your fill in there. Randomness is the opposite of all that. Randomness is like rolling dice. You never know what you're going to get. You just keep repeatedly rolling dice. That's randomness. Or it's like continually selecting a different card from a deck of cards. That's randomness. Or it's like telling a computer programmer to try to create a program by just randomly selecting the keys that they use. Since we know ahead of time that a computer program requires code, if a programmer just randomly enters figures, hitting keys, you are not going to get a program, are you? That's what randomness is like. Randomness then would result in the bonds being used in DNA in a random fashion. If randomness created DNA, there would be a complete disarray of bond usage. There would be no structure, no order, no logic to it, no pattern. And if that happened, DNA couldn't function, couldn't exist, and life could not exist. So randomness, which is the basis of evolution, doesn't have the ability to create all those things that we see in DNA. All the order, the logic, the pattern structures. But guess what? It's not just the bond patterns. In order to have those bonds, you have to have all the atoms in the right place. Therefore, life depends on the correct placement, order, choreography, patterns, etc. of each atom in DNA that is necessary to create each of the types of bonds. Which also means the nature of the hydrogen bonds used in DNA is completely dependent on the chemical structure of each nucleotide, the building blocks that go into DNA. And that's one of the reasons why I explained the composition of those to you a couple weeks ago. You have to have the exact atoms exactly as they are in the structure. And you have to have the exact atomic structure necessary to be able to create the hydrogen bonds in the first place, because what you need to know is most atoms cannot form hydrogen bonds. So once again, we find that both intelligence and prescience are absolute requirements for and during the creation of DNA, because we again see a pattern to which atoms end up in the appropriate position to form the hydrogen bonds. And once again, we see aspects of the design inference. Here we go. Therefore, you have to know the structure of each nucleotide and which atoms have the capability of forming hydrogen bonds ahead of time. You have to know the exact structure of a nucleotide ahead of time. Exactly where the hydrogen, nitrogen, and oxygen atoms must be in each structure of the nucleotide in order to be able to plan to strategically, intentionally, and intelligently use them in the design and the creation of DNA. So that when the two strands come together, they can form the hydrogen bonds in the middle that hold them together. And once again, you don't get any of that from randomness, the hallmark of evolution. Randomness would result in atoms being used in DNA in, guess what? A random fashion. If randomness or evolution created DNA, there would be a complete disarray of atom usage. There would not be any structure, order, logic, or pattern to it. Such a disarray of atom usage would prevent the possibility of organized, ordered, logical, intelligent, life-essential patterns in the hydrogen bond usage that's found in DNA. That would result in a change in the nature and the composition of the structure of DNA, and it would no longer be allowed or capable of performing its life-essential functions. The result of all that would be that life would not be possible. So randomness doesn't have the ability to create any of this. None of the intelligence, the order, the choreography, the patterns of atom usage that we see in the structure of DNA. The structure of the the structure of the nucleotides, as well as DNA as a whole, also have to be exactly as they are in order to bring each atom together in space that can form a hydrogen bond. When the two strands come together, the backbones come together, and then the bases in the middle that form the rungs, like we said, it ends up looking like a ladder, like we showed on that 2D image. When that comes together, there's only so much room on the inside. If you have bases that are too large, they wouldn't fit together. The whole thing wouldn't come together and fit. Wouldn't give the orientation necessary for the hydrogen bonds to be created. If you had bases that were too small, they would fit in there, but they wouldn't bring the atoms necessary to form the hydrogen bonds together in the positions that they need to be in. They wouldn't reach one another. That's also a problem. So being too big or too small is also a problem. Any base structure at all that doesn't bring the required atoms in the place they need to be to form hydrogen bonds will not allow the hydrogen bonds to occur. So what that means is that the hydrogen bonds have to be as they are spatially in order for DNA to be able to open and close and yet remain intact and unchanged. So you have to know all these space requirements ahead of time, which again means intelligence and prescience have to be involved. And again, the good analogy of all this, once again, is the zipper. If the teeth of a zipper, and I actually looked up the parts of a zipper. They're actually called the teeth. That's what they're called. The little sliding thing is the slide. The tab that sticks off of that you pull is called the pull tab. I learned all that this week. If you had the teeth in varying sizes, would a zipper work? No, it wouldn't. It wouldn't work. It would never be able to open and close because they would physically not allow it to happen. And that's the point we're getting at. DNA is exactly the same way. And when you look at a zipper, you can see that a zipper was designed to be open and closed in just the way it does. It's the exact same way with DNA. And again, with this, we see another aspect of the design inference. You have to know the structure and functions of the finished product, the DNA, before you can even start creating the structure of the building blocks. You have to be able to see all of that ahead of time. But for evolution to do this, there we go, randomness now needs to see the finished product before it even forms the building blocks. But as I've been pointing out, randomness doesn't have the capability to do that because it's random. It's not strategic, it's not planned, it's not ordered, it's not logical. There's no patterns, nor does it have foresight, which is exactly what's required here. Now, without the exact pattern of hydrogen bonding that we see between the bases, where A always binds with T, I showed you, G always binds with C. DNA is no longer a blueprint. It's no longer a template. And without that, life can't exist. Without that, DNA is no longer logical, comprehensible, or reproducible. It's no longer intelligent or usable, so life can't exist. So without that pattern of binding between A and T and G with C, every time you try to read a stretch of DNA, which needs to be done when you copy it or when you start trying to make a protein from its code, which we're going to talk about next week, every time you try to do that, you'd get a different result. You could never make an identical copy of DNA. You could never make the same protein. So DNA could no longer function. And if it can't function, if it's no longer reproducible, the fallout of that is that life ceases. So you have to know that pattern of bonding, A with T, G with C. You have to know that ahead of time to be able to strategically, intentionally, and intelligently use DNA in all its functions as a blueprint and a template. And furthermore, let me show you this again. Here's that image. This is what we were talking about. 2 hydrogen bonds between A and T, 3 between G and C. That's the pattern, A with T, G with C. But because there's 3 bonds between G and C and only 2 between A and T, it stands to reason, and yes, it's true, that the bonds between G and C end up being a stronger bond than A and T because they have an extra bond. And that's why A's and T's are found in places where DNA needs to be opened up, like where it needs to be copied, or where the start areas are for where it codes for how to make a protein. You find a lot of A's and T's there because they're not as strong of a bond. However, G's and C's are found in places in DNA where much greater scrutiny and regulation is required. So once again, we see a pattern of when to use which bases. And that's been designed into the overall structure of DNA as well as in the subregions that I was just mentioning. So you not only need to know the pattern of binding, you need to know when to use which bases. And you have to know that again ahead of time in order to strategically and intentionally use them exactly as required in the design of DNA. That's actually repeated. Without all that, without using that pattern, every time you try to copy DNA or try to make a protein from its code, you'd get a different result. And if that happens, life ceases. So the reproducibility in DNA is absolutely essential for life. Now, another thing we can talk about with all this is where the energy comes from that powers everything that DNA does and the proteins that work on DNA. And the energy is found right here in this bond. It's found in the phosphate bonds of a nucleotide. A free nucleotide, this is a building block. Remember, the nucleotides are the building blocks of DNA. A free nucleotide can have 3 phosphate groups. 1, 2, 3. It can have 3 of them. That's why this is called triphosphate, adenosine triphosphate. But when it goes into DNA, look what happens. In the backbone, there's only 1. That's because the other 2 will get removed before it's added to the growing strand. And breaking that bond, it's called a phosphodiester bond. I give you that free, I won't charge you any extra for it. Breaking that bond creates all of the energy. There it is right there, the phosphodiester bond. That's where we get the energy from breaking those bonds to perform the functions that DNA does and provide the energy for the proteins that work on and in and around DNA from that bond. Therefore, you have to know the energy requirements required for DNA functions and the proteins that work on it, and the fact that such a bond there can provide that energy demand. You have to know that ahead of time in order to strategically, intentionally, and intelligently use these bonds in the structure of the nucleotide exactly as required. And remember when I said, without all the nucleotides lining up the way they do in the DNA backbone, DNA wouldn't have directionality. And again, that's what that looks like. The way this is drawn, this strand you would say is going up. The left strand over here is going down. Remember, both strands, each one runs in opposite direction of the other one. So, without this, the proteins that move along DNA when it's doing their functions, like when it's copying it or when they're reading it to start making a protein, they would never know which direction to read the DNA. Because if you latch onto a DNA strand, there's two ways you can go. They need to know which is the correct way to go. But without that, they would never know which direction to read the DNA. That would be a huge problem. Because depending on which direction you go, you'll get completely different results. You would never get the same results twice, and guess what would happen there again? Life could not exist. Life would cease. So, again, you have to know the directionality created by each nucleotide and each strand. You have to know that ahead of time to design them exactly the way they are in the structure of DNA, and so the proteins knowing that function with DNA can perform their functions the way that they're supposed to. Here's another one. We talked about all the extensive packaging and folding that DNA goes through. If it didn't do that, you would never be able to house the DNA inside of a cell. If you remember, I said each of our cells has about 6.5 feet of DNA. If you took it all and laid it out end to end, you have to get all that inside the cell. If you didn't, DNA would be vulnerable to things like degradation. It's going to get chewed up, and it's going to easily get destroyed. So, all those proteins that are involved in this, like the histones were ones that we mentioned, and the capabilities within the structure of DNA itself that allows it to be folded up, they are all necessary for DNA to exist and for life to exist. But you not only have to get it in there, remember, in order to get at it and use it, you have to unwind it, open it up, access its information, all doing that without destroying this. So you need to know how to package and condense DNA to fit inside a cell in a way that it can still be opened up and accessed without being destroyed. You have to know that ahead of time. You need to have a plan for how that's going to happen ahead of time in order to strategically, intentionally, and intelligently fold the DNA, condense it exactly as needed, and still allow it to be unfolded and accessed without getting destroyed whenever it needs to be used. And there's one point I want to finish with today, one more point. Nucleotides have handedness, meaning they're either right-handed or left-handed, okay? And although this isn't the whole story, for analogy purposes, you can imagine right-handed and left-handed nucleotides being mirror images of one another. Sort of like your right hand and left hand, but your right and left hands aren't really mirror images of one another. If you look at them closely, they're not exactly the same. But for these purposes, we're going to view them as being mirror images of one another, where they're chemically equivalent, but they're different spatially. And actually, I'll give you something else free of charge. The way they determine it is they pass light through it, and the light is polarized, and they determine whether it comes through, whether it corkscrews right, clockwise, or counterclockwise. That's how they actually determine the difference between right-handed and left-handed. But right now, we're just going to go right-handed, left-handed, okay? Now, nucleotides almost always form in living organisms. And when they do, they're right-handed. Only on rare occasions do they form outside of life. But if they ever do, they contain an even mixture of both right-handed and left-handed nucleotides. And what you must know, there is no natural mechanism in existence to screen out the left-handed forms. Folks, this creates a problem for evolution. Let me explain it to you. If the first nucleotides form randomly outside of life, which they would have had to, there would have been an even mixture of both right-handed and left-handed nucleotides. However, since only the right-handed ones are used in life, how could their incorporation into DNA been regulated to only include the right-handed ones when there is no mechanism in existence to screen out the left-handed ones? The fact is, there would not have been a discriminatory use of nucleotides. Both forms would have been incorporated into DNA from day one. But as we know, this is not the case. Only right-handed nucleotides are incorporated into DNA. And as I said, that creates a big problem for DNA, for evolution. But guess what? It only gets worse because it doesn't stop with the nucleotides. It's the same story for amino acids, which are the building blocks of proteins. When amino acids are synthesized, are found outside of life, they too come in an even mixture of chemically equivalent forms. Right-handed and left-handed amino acids. But once again, in life, amino acids are left-handed, with the exception of sometimes being found right-handed in diseased or dying tissue. So when you have them as right-handed, their presence is not a good thing. And just like with nucleotides, no process exists, no natural process exists that can isolate or screen out the right-handed or left-handed versions of amino acids. And once again, we have the same problem for evolution. If the first amino acids formed randomly outside of life, which they would have had to, there would have been an even mixture of both right-handed and left-handed amino acids. However, since only the left-handed ones are used in life, how could their incorporation into proteins been regulated to include only the left-handed ones when no mechanism was in place to screen out the right-handed ones? The fact is, there would not have been a discriminatory use of amino acids. Both forms would have been incorporated into viable proteins from day one. But as we know, this is not the case. Only left-handed amino acids are incorporated into viable proteins. As I said, this is another problem for evolution, but guess what? Doesn't stop there either. It's the same story for sugars. As with nucleotides, sugars are all right-handed in life. But if they came about randomly, where's all the left-handed ones in life? And you know, you can't even eat left-handed sugars because your body will not process them. The same way I was talking about before, about determining by passing light through and seeing which way. Tell you what, we'll talk, oh, I'm about done here. We'll talk about that next, okay? So, once again, we have a problem for evolution. Because evolution simply cannot explain any of these with the nucleotides, with the amino acids, or with the sugars, how there's a discriminatory use of them. And we could go on like this for a long, long time. We could talk about meiosis and mitosis, all the structures involved there. We could talk about how DNA gets divided up into chromosomes. We could do this all day, but we won't. I'll spare you. Because we're going to stop there on this subject today. We're going to end there. And next week, we're going to talk about some of the actual functions of DNA and then some of the evidence that we get from that. So we will close there today.